Once you have the reference sequence, you can start the mapping. And for mapping RNA-seq reads, there is more choice in mappers than for mapping DNA-seq reads. So these are the most important ones that I listed here. So you have STAR, HiSET2, Callisto, and Salmon. So what are the good things about mapping to the genome, like STAR and HiSET2? Well, the good thing there is that you will find things that are not annotated. If reads are mapping in an intergenic, it means that there is a transcript there that has not yet been annotated. Yeah? You will find these reads and these transcripts, these novel transcripts. So you will find novel genes, you will find novel isoforms. What is the difference between these two? Well, they do more or less the same thing. There's one difference. It's how they handle the genome sequence. So STAR tries to put the entire genome sequence in the memory of your computer. So for human and mouse, this is tricky, of course. It's huge FASTA file that he tries to fit in the memory of your computer. So he needs a lot of memory to work. And on a regular laptop, it will be very slow. HiSET chops the genome into smaller pieces. He generates smaller indices, so he can fit the genome in smaller parts in the memory of your computer. As a result, HiSET can be easily done on a laptop. Okay. Both tools are in Galaxy. And in Galaxy, there's no issue with memory. Galaxy has memory enough. What is the benefit of mapping to the transcriptome instead of to the genome? Well, mapping to the transcriptome makes it really easy to quantify known splice variants. So if they are known, then these tools can make a distinction between different isoforms. And they can say for each splice variant separately, well, this is the number of reads that maps here. These two tools cannot do that. Yeah? They will give you number of reads that map to a gene, all isoforms included. That's what the output here will be. So if you're really interested in looking at which isoforms are in this set of conditions and which isoforms are used in that set of conditions, then you're better off doing this. Note again, you will not find isoforms that have not been annotated. Okay, so and the two tools here, Callisto and Salmon, how are they different? Well, almost not. They are very, very, very alike. So typically, depending on what you want, you choose one of these two methods, and then which of the two tools you use is not really relevant because they generate more or less the same output. So how do you choose? Well, when you're interested in differentially expressed genes, mainly, you go for this. When you're interested in novel genes and novel isoforms, you go for this. When you're interested in splice variants, specifically, not novel ones, but known ones, then you have to go for this. So it depends on what you're interested in. Now, what is the plus here? The plus is that there are ways to use the Callisto and Salmon output that is specific for isoforms, for transcripts, to transform that output into gene level counts. So you get a similar output as for these. So basically, for this, you can only look at differentially expressed genes. And with these tools, you can do both. You can look at differentially expressed genes, and you can look at differential isoform usage. Yeah? But novel things will only be found by mapping on the genome. All of them are in Galaxy.